Welcome back to the deep dive. We talk a lot about the brain's ability to learn its plasticity.
All the time.
But today, we're going a layer deeper. We're looking at the fundamental organization of the motor cortex before all that extensive learning happens.
We're looking at the factory settings.
The factory settings, I love that. So what are we zeroing in on?
We're looking at a study on the anterior lateral motor cortex, or ALM, in mice. And this is so critical because, almost every study before this looked at animals that were already experts at a task.
So their brains were already molded by that training.
Exactly. Their neural patterns were set. The goal here was to sort of reverse engineer it, to see the blueprint of connectivity before learning really takes over.
But this gets at a huge question, right? How does the brain even decide how to represent a movement goal? Is it all just top-down commands from other brain areas?
Or is it the local network, the chatter between the neurons right there that really shapes everything?
And to figure this out, they had to be pretty clever with the setup.
Oh, incredibly. They use a natural behavior mice reaching with their tongues, which they already know how to do so. No complex training was needed. And they mapped it all with this revolutionary all-optical method.
So our mission for this deep dive is to unpack what they found. We'll get into how this untrained brain encodes goals and outcomes, how it's all organized physically, and then this really fascinating discovery of these rare but critical hub neurons.
The coordinators of the whole system. It's a fantastic story.
Okay, let's start with the behavior itself. You said it was natural. The mice are head restrained, but they're just licking to get a reward.
That's right. They're reaching with their tongue for a drop of water. But the target could appear at up to 25 different spots on a grid.
Wow, so it's not just one simple target.
No, it's a wide space. And because it's a natural action, they engage with it almost immediately. This gives us a really clean baseline to see how the ALM works from the get-go.
And while they're doing this, you've got researchers looking directly into their brains. How are they measuring the activity of these neurons?
They're using something called volumetric calcium imaging.
Right, with GCAM-P6s.
Precisely. You can think of GCAM-P as a tiny little biological camera inside each neuron.
So when the neuron fires, it lights up.
It glows. Calcium floods the cell when it fires, and the GCAM-P protein lights up. They measure that change in fluorescence, they call it FF, and then they can mathematically work backward to estimate the neuron's actual spike rate.
Okay, so they have this incredibly detailed view of firing patterns. What was the first big shocker they found, just in terms of timing?
Well, this is where it really challenges what we thought we know from all those studies on trained animals. Nearly 80% of the neurons they imaged were active during the task. But, and this is the key, the activity usually peaked after the mouse had already started moving.
After.
Sometimes seconds after.
Wait a minute, I thought the motor cortex was the command center. It says, go. Why would it be most active after the tongue is already out there?
It's totally counterintuitive, isn't it? In a highly trained animal, you'd see the peak before or during the action. That's the planning and execution signal.
Right.
This post-movement activity in a naive brain, it tells us something really fundamental. At this early stage, the ALM isn't just about planning. It's heavily, heavily focused on processing the consequence of the action.
It's evaluating what just happened.
What just happened and where are we trying to go? It's holding that information online.
So to evaluate, it needs to know the goal. Were the neurons tuned to specific locations on that grid?
They were. About 23% of them were tuned to a specific target location. They had what's called unimodal tuning.
Meaning one preferred spot.
One preferred spot, yeah.
Okay.
And altogether, these neurons tiled the entire grid. They did see a slight bias for the corners, maybe because those are harder to reach.
Okay, so we have the where, the location, and we have the when, that surprising post-movement timing. Did they see neurons that did both at the same time?
Oh, massively. They called it conjunctive encoding. Almost 90% of those location-tuned neurons also had this specific temporal tuning.
And was that post-movement thing still there?
It was. Get this. Almost a third of these conjunctive cells were firing 2 to 6 seconds after the lick was already over.
So that neuron isn't just saying go to X, it's holding a memory. It's saying, we just went for location X, and I'm still thinking about it.
That's the perfect way to describe it. It's the raw material for learning. You're linking the outcome to the goal that caused it.
Okay, let's talk about that outcome, the reward.
They manipulated it right.
They did. On most trials, it was a standard reward. But on 10% of trials, they'd randomly triple the amount of water,
A jackpot.
a jackpot, and on another 10%, they'd randomly omit the reward completely.
The rug pull.
Exactly. And they found huge populations of neurons that cared about this. Nearly 20% responded to the increased reward, and 10% responded to the omission. But the real magic, the thing that ties it all together, is finding neurons that care about both the location and the reward, isn't it?
That's the absolute key. About 8% of all the ALM neurons they recorded showed conjunctive tuning to both the target location and the reward outcome. This is the neural signature of a specific action linked to a specific result.
So how did the reward change what the neuron was doing? Did getting a jackpot make a neuron suddenly prefer a different location?
No, and this is so elegant. It didn't change the preference. It acted as a gain modulation.
Okay, unpack that. Gain modulation.
Imagine your neuron is a radio tuned to its favorite station. Let's say that's location X. When the reward increases, it's like someone turned the volume knob way up. The neuron fires much more intensely, but the station it's tuned to, location X, doesn't change.
So it changes the amplitude of the signal, not the signal itself.
Precisely. And that distinction is everything for learning.
Why? Why is that so important?
Because it delivers what's called vector feedback, not scalar feedback.
Okay, vector versus scalar. Give me an analogy.
All right. Scalar feedback is getting a year-end review that just says, good job. It's nice, but not very useful for improving.
Vague.
Very vague. Vector feedback is your boss saying, you excelled on project X because you used strategy Y. We need more of that.
It's specific, actionable.
It's a vector. It points directly to the policy you need to reinforce. That's what this conjunctive encoding does. It tells the brain circuit which specific action led to a good outcome, allowing for incredibly rapid, efficient learning.
Okay, let's pause and recap this first part. It's a lot to take in.
For sure. So first, in the untrained brain, ALM motor cortex activity peaks after the movement. It's focused on evaluating the consequences.
And it encodes that information with what you called vector feedback. It combines the target location and the reward outcome into a single signal that's perfect for fast learning.
And finally, this isn't just a random salt and pepper mix of neurons. Cells with similar jobs, similar tuning, are physically clustered together. They form functional columns.
Which brings us to a really big question for you to think about. If the brain is already this organized functionally, with these columns and this very specific encoding, how in the world is it actually wired up to support that?
How do you build a circuit that does all this? You need a map.
You need a map. And that brings us to the method. Measuring connectivity, who's talking to whom while an animal is behaving. That's been the holy grail for a long time.
A huge challenge. Most methods require you to disrupt the very circuit you're trying to study.
So what was the breakthrough here?
The all-optical method. It is a genuine landmark achievement. It lets you manipulate neurons and record the results at the same time in the same volume in a behaving animal.
Okay, break that down. All optical. What two technologies are they combining?
So they combine two-photon optogenetic stimulation with that volumetric calcium imaging we already talked about.
So the imaging is the recording part. What's the stimulation part?
For stimulation, they use a protein called crimson R. It's an opsin, which means it makes a neuron sensitive to light.
So they can use a laser to basically flip a switch on a single neuron and make it fire on command.
On command. A tiny, focused, two-photon laser hits one target neuron, activates the crimson R, and makes it fire. And at the exact same time, using the GCAMP signal, they're watching thousands of its neighbors in 3D to see who responds.
They're literally poking one neuron and listening for the echo across the network.
That's a perfect way to put it. And it lets them map causal connectivity. Not just who is sitting next to whom, but whose firing actually causes a response in another neuron.
The scale of this is just mind-boggling.
Yeah.
Over 22 million possible connections mapped in a single session.
In about 30 minutes, yeah. It's like generating a microscopic wiring diagram of the brain on the fly. And they were super careful with their controls, too.
How so?
Well, they defined their target neurons, the ones they zapped, and the causally connected neurons that responded. But they also zapped other neurons nearby that they knew didn't have the crimson R protein.
A placebo zap.
A placebo zap, just to prove that the responses they were seeing weren't some artifact of, light scattering around. They showed those off-target effects were tiny.
So they have this incredible high-resolution 3D map of the wiring. What did it look like? Was it just a tangled mess of spaghetti?
Not at all. There was a very precise, very predictable structure, a beautiful organization that actually confirmed what they suspected from the functional data.
Okay, so what is this structure?
Well, if you look at a neuron, it is most likely to excite its immediate neighbors. That excitatory connection is strongest locally, and it drops off really fast within about 100 micrometers.
So that's the core of the connection.
That's the goal. But then if you look a bit further out between 100 and 250 micrometers away, inhibition starts to dominate.
Ah, so it's a core of go signals surrounded by a ring of stop signals.
You've got it. And that pattern has a classic name in neuroscience. It's called the Mexican hat profile.
That's a great visual. So you have the tall peak of excitation in the middle, like the crown of the sombrero.
And then the wide, flat brim of inhibition all around it.
What's the functional point of a structure like that? Why build a circuit with an excitatory core and an inhibitory surround?
It's all about sharpening the signal. That inhibitory brim prevents the activity from just spreading out everywhere. It keeps the message contained and ensures that local cluster of excitatory neurons, that column, maintains its specific functional identity.
And this gets us right back to the columns you mentioned earlier.
It does. They found the physical connections perfectly mirrored the functional organization.
How so?
They saw that connectivity went way further down axially into the deeper layers of the cortex. It peaked about 60 micrometers below and extended even further. But laterally across the surface, it dropped off super fast.
So the connections are organized like long, thin pillars running through the brain tissue.
Exactly, anatomical columns. The physical wiring is set up in these vertical units, which provides the perfect scaffold for the functional columns of tuning they observed in the first place.
Okay, time for our second recap. The all-optical method gave us this incredible snapshot of the brain's wiring diagram.
And that diagram revealed a clear structure. The Mexican hat profile. Local excitation, surround inhibition. It's a design built for sharpening signals.
And this physical structure creates anatomical columns, which perfectly match the functional columns they found for encoding goals and outcomes.
Right. So we know similar functions are clustered together, and we know local excitation is really strong. So here's the next question for you to ponder. How does the brain make sure that only neurons with similar jobs get linked in that excitatory core? Is there a specific rule for who gets to connect to whom?
That is the deepest question, isn't it? Does the wiring actually predict the function? Do all the neurons that like location X only talk to other location X neurons?
And the answer was a clear and resounding yes. They found that the stronger the causal connection between two neurons, the more similar their location tuning was.
So the wiring diagram is not random at all. It's exquisitely fine-tuned.
It's fine-tuned. And this relationship was strongest right where you'd expect it, inside that 100-micrometer excitatory core. This is the proof that functional columns don't just happen because of some shared input from far away. They are built and maintained by functionally specific local recurrence.
Like excitatory connections.
The circuit is fundamentally hardwired to strengthen the bonds between cells that do the same job.
Okay, that makes perfect sense. But when they looked at the overall network map, the connections weren't evenly distributed, were they?
Not even close. The distribution of outgoing connections, what they call outdegree, was heavy-tailed.
What does that mean, heavy-tailed?
Well, imagine a random network. In a random network, everybody has roughly the same number of friends. But a heavy-tailed network is like a real-life social network.
Where most people have a few friends.
And then there are a few super connectors, the influencers, with thousands of connections. They have a disproportionate effect on the whole network.
And they found those in the brain.
They found those in the brain. These rare, highly connected neurons are called hub neurons.
And these hubs, they're not just influential, they're also part of a kind of exclusive club, right?
They are. They tend to have high in degrees as well. They receive a lot of connections. It's a rich club phenomenon. They talk to a lot of neurons, and a lot of neurons talk back to them.
But here's the part that I just find so fascinating. Functionally, these big shot hub neurons, they were actually worse at the task.
It's a total paradox. They showed weaker tuning to specific locations and weaker modulation by reward. They were less specialized than their neighbors.
So the most connected, most influential neurons are the least specific. How is that not just introducing noise into the system?
Because their job isn't to encode a specific location. Their job is to coordinate.
To coordinate.
Yes. Despite their weak tuning, the activity of a hub neuron was more correlated with the average activity of its local neighborhood. They act as local coordinators.
You mean analogy for that?
Think of an orchestra. The highly tuned non-hub neurons are the first chair violins. They are playing the beautiful, precise melody that's the specific location information.
Okay.
The hug neuron, it's the conductor, or maybe the entire rhythm section. It's not playing the main melody, but it's providing the beat, the tempo, the structure that allows all the violins to play their specific parts together in harmony.
So the hubs provide the stability that lets the other cells specialize. And they confirm this with modeling.
They did. They built a network model that included all their experimental findings, the spatial clustering, the like-to-like connections, and these rare, weakly tuned hubs. And the simulation showed that the hubs provide this broad, weak, organizing signal to all the neurons around them.
That constant hum in the background.
It's a constant hum that actually enhances the tuning of the specialized non-hub neurons. It stabilizes the bump of activity that represents the target location. So the hubs don't encode information themselves, they amplify the network's ability to encode information cleanly.
Wow. Okay, so let's try to put this all together. This work has really redefined our baseline understanding of the motor cortex.
It absolutely has. The ALM isn't a blank slate waiting for instructions. It's a highly structured network from the start with a columnar Mexican hat architecture.
And that architecture is built for one thing: efficient learning. It combines location and outcome to provide that vector feedback you were talking about.
That targeted, rapid learning signal. And the physical wiring that supports it is this beautiful two-part system. You have the like-to-like connections for specificity.
But the whole thing is orchestrated by these rare, influential, but weakly tuned hub neurons.
And being able to map the causal links while seeing the function proved that it's these local recurrent connections that really drive the whole organization. This is the neural substrate, ready and waiting for learning to happen.
Which leaves us with a final, really provocative thought for you to take away. We know these hubs are coordinators, less tuned to the specifics of any one task. If they're there to organize the network for any potential learning, does that mean the hubs themselves represent a kind of innate, genetically hardwired framework for motor control?
While the more specialized, highly tuned neurons are the plastic parts ready to be sculpted by experience. It could be the fundamental division between the brain's innate structure and its learned skills.
That's the next layer of complexity to explore.
And that's where the ALM goes from its factory settings to a truly personalized machine. Thank you for joining us for this deep dive.
We hope you enjoyed this look at the cutting edge of motor cortex connectivity. We'll see you next time.